It is just a 9 amino acid sequence that is derived from the viral proteins presented on the surface of the cell. That peptide epitope is recognized by a T cell. That's where all the action happens. Once the T cell, as shown in this picture, recognizes the viral epitope on the surface of the target cell, then it will try to kill that cell. So what does the HIV virus do? But it changes its epitopes. And that's actually shown on this slide, what happens in the course of a single person's infection. If you follow the sequence of the HIV virus from the point that they're infected to just several years later, you can see that their virus evolves, and it evolves variants with mutant epitopes that escape the immune response. This is a picture of the sequence of a child infected in Philadelphia at birth. And only 4 years later, you can see how many different sequences of HIV are circulating in this child's body. This has also happened worldwide. What happens in a child happens in a population. So within a population, HIV again is trying to escape the immune responses of the collective population. The result is that in different regions of the world, different variants of HIV have emerged. These are called clades. In the United States, we have clade B. In Europe, clade B. In Africa, which is the epicenter of the epidemic, we have 5, 6, 10 different clades. In a single country, Mali, where I work, the clades are mixed, and you have both the A and the G clade circulating in the population. This is a dramatic problem in terms of making an HIV vaccine, because you can't make a vanilla vaccine and expect it to protect against chocolate. It simply isn't going to work. This picture actually shows how great the variation of the HIV virus has been compared to one of the viruses that we are most afraid of, which is the flu virus, shown over in the corner of the picture right here. I don't have a pointer, do I? I don't think so. But you can see there's a little dot over in the corner of the picture over here, and that shows you how variable the flu virus is in the course of a year compared to the expanding sequences of HIV. When we really think about this, it's amazing that HIV can hold itself together. How can it be so variable and still function as the same virus? So this is really summarizes the immunopathogenesis, the generation of the bad immune response to HIV from the vaccine perspective. First of all, the near impossible task of preventing HIV entry makes it difficult to make the most standard type of vaccine against HIV, which would be an antibody-directed vaccine. The HIV variability is also a huge problem, because if the immune system must recognize those epitopes that are presented on the surface of the target cell and the virus mutates the epitopes, the T cells can't keep up. They can't keep on recognizing an HIV that is mutating away from their immune response. The other problem is latency. You may be able to clear HIV infection with drugs in the body, but there's always the coding sequence for HIV hiding inside of a cell, waiting for the cell to be activated. So you can't clear HIV. It's very difficult. The other problem with HIV is that it destroys the very immune system that we use to fight it off. So the T cell, which is the target of HIV infection, is also the cell that is destroyed and leads to the disease that is known as AIDS. So those are all huge problems in terms of HIV and vaccine development. So what do we think we can possibly do? There are ways that we could perhaps develop a vaccine, and that's what I'm going to get into right now. We're thinking that one of the ways that we could possibly direct our HIV vaccine effort is to prevent infection. And one of the greatest problems with that is that most of our preventive vaccines, like the hepatitis B vaccine or the cervical cancer vaccine that you've heard about, are based on antibody response. Most vaccines that prevent infection are based on antibody. So it's difficult to conceive of an HIV vaccine that could actually effectively prevent infection. So now we're thinking perhaps a vaccine might be able to prevent disease. meaning, yes, you still get infected, but then we're able to control the infection, and we turn you into a person who has HIV infection, but maybe like having diabetes or high blood pressure, you live with the disease rather than being killed by it. The other thing that is a potential goal for a vaccine is preventing secondary transmission, so preventing transmission from one person to another by lowering the amount of the virus so it doesn't get transmitted during sexual intercourse. So let's talk a little bit about the different types of vaccines, and then I'm going to talk about why the Merck vaccine has been such a failure. The ways that we can work on vaccines are shown here, and I'm just going to first talk about the simplest approach to making an HIV vaccine, which is what scientists seized upon in the 1980s when the virus was first identified. And that is, if you look at the surface of this virus, you can see a knob on the surface that's called the envelope protein. When people saw that it had an envelope protein, they thought, this is just like hepatitis B. We can simply sequence that protein, create a whole lot of it in a big vat, make just tons and tons of this particular protein called GP120, and we can inject that into people, and we will create an effective antibody response. By now, you should be familiar with the two things that absolutely make that impossible. So one is the variation of the virus. This particular envelope protein is the most variant protein in the whole virus. So maybe it's not so simple to select one single protein that will be effective against all strains of HIV. That would be problem number one. And problem number two is that nobody's been able to figure out what antibody can effectively prevent entry into the cell. So there are the two problems with that approach. Nonetheless, as Pat Thomas says in her book, Hot Shots, nonetheless, even though most scientists were well aware that that approach would not work for HIV, vaccines were developed, trials went forward, people volunteered all over the world for the VaxGen trial that came off about 2 years ago, and were we surprised that the results were that there was absolutely no protective effect? Most of the scientists in the field were not surprised at all. VaxGen went on to capture some defense money and is now making smallpox vaccines somewhere on the West Coast, but that particular vaccine effort completely failed. At that point, what do we do as scientists? We go, well, maybe that didn't work, we'll try something else. So two of the leaders of the NIH vaccine effort, Peggy Johnson, a good friend of mine, and Tony Fauci, who's the director of the Division of AIDS, decided that perhaps we would have to think about a different way of making an HIV vaccine. Maybe a less than perfect vaccine would be okay. Maybe something that didn't completely prevent infection but actually diminished disease would be something that would be acceptable. And the reason for this is that we know that some people can actually control HIV infection. There are people who are living with HIV today, 20 years after being infected, who have never been ill. Something about their body allows them to contain the HIV infection, and so people started looking into that. They measured the amount of virus in their blood, and they saw that if you had a low load or viral load of HIV virus, then you were a controller, somebody who was an elite controller, they're called. And this is what people started to study. They found, lo and behold, that probably the most important correlate of protection from disease was T-cell mediated immune response. The T cells, those same T cells recognizing epitopes, sometimes were able to keep the virus in check. So people started working on T-cell mediated vaccines instead of